Hello, dear students. This lesson is entitled Passive and Active Transports. By the end of this lesson, you should be able to explain how cell membrane structure relates to its function, describe both passive and active transport mechanisms, and discuss how these processes are applied in real-life biological situations. As we proceed, consider these essential questions. How do cells maintain stability despite changing environments? The cell membrane acts as a semi-permeable barrier, regulating what enters and exits the cell. Also known as the plasma membrane, it encloses the cell and protects its internal environment. This diagram shows a typical animal cell. Notice that the cell membrane forms the outer boundary of the cell. Inside, you can see organelles such as the nucleus, mitochondria, ribosomes, and endoplasmic reticulum. All of these structures depend on the membrane to control what enters and leaves the cell. The membrane protects the internal environment while allowing communication with the outside. This diagram represents a plant cell. In addition to the plasma membrane, plant cells have a cell wall, which provides structural support. The plasma membrane is located just inside the cell wall. This image represents the fluid mosaic model of the cell membrane. The term fluid refers to the lateral movement of phospholipids and proteins within the membrane. The term mosaic refers to the patchwork arrangement of different proteins embedded in the bilayer. These proteins serve various functions, such as transport, signaling, and cell recognition. This diagram illustrates how amphipathic phospholipids arrange themselves to form a bilayer. Each phospholipid has a polar, hydrophilic head that is attracted to water and nonpolar, hydrophobic tails that repel water. As a result, the heads face the aqueous environments inside and outside the cell, while the tails face inward. This arrangement creates a stable barrier between the cell and its surroundings. Cellular transport refers to how substances move across the cell membrane. This process allows cells to take in nutrients, release wastes, and maintain internal balance. This diagram provides an overview of two transport mechanisms in cells, passive and active transport. Let us first look at passive transport, which does not require energy and moves substances down their concentration gradient. Diffusion is the movement of molecules from an area of higher concentration to an area of lower concentration. Movement from high to low concentration is also called movement down or along a concentration gradient. This movement continues until equilibrium is reached. This diagram visually represents diffusion. Notice how purple dye crystal particles spread out evenly over time. Initially, these particles are concentrated in one area. As time passes, they move randomly until they are evenly distributed. Diffusion does not require energy because the movement is spontaneous. Facilitated diffusion is a type of passive transport that allows substances to cross the cell membrane with the help of specific transport proteins. Like simple diffusion, it does not require energy in the form of ATP or adenosine triphosphate because substances move down their concentration gradient from an area of higher concentration to an area of lower concentration. Osmosis is the diffusion of water through a selectively permeable membrane. In this diagram, the fissile tube on the left contains a solution with a higher solute concentration, 10%, compared to the surrounding beaker solution, 5%. Because the membrane allows water to pass but not solute, water moves from the region with more free water, or lower solute concentration, in the beaker into the tube, where there is less free water, or higher solute concentration. Over time, this causes the water level inside the tube to rise, as shown on the right. The central panel illustrates this at the particle level. Water molecules move in both directions, but there is a net movement of water toward the side with higher solute concentration. This process is crucial in biology because it explains how water is distributed among cells, tissues, and body fluids, helping maintain proper cell volume and overall homeostasis. This diagram compares how animal and plant cells respond to hypotonic, isotonic, and hypertonic solutions. In a hypotonic solution, water enters the cell. An animal cell swells and may burst because it lacks a cell wall. A plant cell becomes turgid, as the rigid cell wall prevents bursting and creates turgor pressure. In an isotonic solution, water movement is balanced. An animal cell maintains its normal shape. A plant cell becomes flaccid, meaning it lacks firmness due to reduced turgor pressure. In a hypertonic solution, water leaves the cell. An animal cell shrinks. A plant cell undergoes plasmolysis, where the membrane pulls away from the cell wall. Overall, plant cells are protected by a cell wall, while animal cells are more vulnerable to changes in their surrounding environment. Under normal conditions, your cells exist in an isotonic state, meaning the solute concentration inside and outside the cell is balanced. Osmoregulation is the process by which the body maintains this balance by controlling water movement. This is how your cells absorb water when it is needed. When athletes drink too much pure water, the fluid outside the cells becomes less salty. As a result, water moves into the cells, causing them to swell and possibly burst. This is why sports drinks contain electrolytes, which help maintain isotonic conditions. The opposite problem occurs when drinking salt water. When salt water is consumed, the concentration outside the cells increases. Water then moves out of the cells, causing them to shrink, even if the body is dehydrated. This explains why drinking seawater worsens dehydration. The next type of transport mechanism is active transport. It is the movement of substances against the concentration gradient, from low to high concentration. This process requires an expenditure of energy, typically from ATP. Many essential nutrients are more concentrated outside the cell than inside. Active transport allows cells to take in these substances. It also allows the removal of toxic wastes, preventing them from building up inside the cell. The diagram on the left shows protein pumps embedded in the membrane. A proton pump is a membrane protein that uses energy, usually from ATP, to actively transport hydrogen ions across a membrane and create an electrochemical gradient. The diagram on the right shows cotransport, a type of membrane transport in which the movement of one substance down its concentration gradient drives the simultaneous transport of another substance across the membrane. Bulk or vesicular transport moves macromolecules like proteins and polysaccharides into or out of the cell. This process requires energy in the form of ATP. There are two types: endocytosis, which brings materials into the cell, and exocytosis, which releases materials from the cell. Endocytosis is a type of active transport, where a cell takes in large molecules or particles by wrapping its membrane around them, forming a vesicle. This energy-requiring process allows cells to absorb substances like nutrients, fluids, or even other cells. Endocytosis includes phagocytosis, pinocytosis, and receptor-mediated endocytosis. In phagocytosis, the cell extends pseudopodia to engulf large particles. The material is enclosed in a food vacuole, which is later digested. The image on the right is a micrograph of an amoeba engulfing a green algal cell, illustrating phagocytosis in action. In pinocytosis, the cell takes in droplets of extracellular fluid. This process happens continually and involves small vesicles. On the right are micrographs, which provide real examples of pinocytotic vesicles forming in the membrane. In receptor-mediated endocytosis, extracellular molecules bind to matching receptor proteins embedded in the plasma membrane. These receptors cluster in regions called coated pits, which are lined with coat proteins that help the membrane curve inward. The membrane then pinches off to form a coated vesicle containing the bound substances. Once inside the cell, the vesicle may release its contents into the cytoplasm or fuse with a lysosome for digestion. Exocytosis is the cellular process where intracellular vesicles fuse with the cell membrane to release their contents into the extracellular space. The Golgi body packages macromolecules into secretory or transport vesicles. Fusion of these vesicles with the plasma membrane spills their contents out of the cell. This diagram of a typical animal cell shows the Golgi apparatus. In a plant cell, the Golgi apparatus also plays a role in preparing materials for transport and secretion outside the cell. This illustration further demonstrates the exocytosis process, where a vesicle fuses with the plasma membrane to release its contents. The diagram shows this membrane fusion step, while the micrograph provides a real cellular image of a vesicle opening to the extracellular space. Cellular transport mechanisms have several real-world applications, including nutrient absorption and drug delivery. This question asks how animals extract nutrients. The small intestine is lined with cells that use passive diffusion, facilitated diffusion, and active transport to move nutrients from digested food into the bloodstream. For example, glucose enters intestinal cells using transport proteins. Other applications include insulin regulation, which uses receptor proteins to allow glucose into the cell. HIV uses facilitated diffusion and receptor-mediated endocytosis to infect host cells. Insulin is a protein hormone that binds to receptor proteins on cell membranes. When insulin binds, it triggers transport proteins to move glucose into the cell. This process helps regulate blood sugar levels. HIV uses a combination of facilitated diffusion and receptor-mediated endocytosis to enter host cells. It binds to specific receptor proteins on the membrane and is taken into the cell in a vesicle. By understanding these transport processes, scientists can develop treatments that block viral entry into human cells. Artificial kidney dialysis is a life-saving treatment that uses diffusion across a semi-permeable membrane to remove waste from the blood, either through peritoneal dialysis performed inside the body or hemodialysis, where blood is cleaned outside the body using dialysate fluid and then returned. Peritoneal dialysis uses the lining of the abdomen as a natural semi-permeable membrane, where dialysate fluid is added to the abdominal cavity. So waste products and excess water diffuse from the blood into the fluid, which is then removed from the body. In hemodialysis, Blood is removed from the body and passed through a dialysis machine where waste products diffuse into a dialysate fluid across a semi-permeable membrane, and the clean blood is then returned to the body. Desalination uses reverse osmosis to remove salt from seawater, applying transport principles industrially. Desalination is especially important in areas with limited freshwater supplies, helping provide clean drinking water and support agriculture and industry. Reverse osmosis is a process that uses pressure to force water across a semi-permeable membrane, allowing water molecules to pass through while removing dissolved salts, minerals, and other impurities. It is widely used in desalination and water purification to produce clean, safe drinking water. To answer the essential question, remember that cells maintain internal stability, or homeostasis, by carefully controlling their internal conditions, even when the external environment changes. This is achieved through membrane transport mechanisms, which regulate the movement of water, nutrients, ions, and wastes into and out of the cell. To conclude, reflect on membrane structure, examples of passive and active transport, and the difference between endocytosis and exocytosis. Thank you for listening.